So this is mitosis part 2, just continuing on. So the next phase is metaphase, and the prefix meta means middle, okay? And what ends up happening is our chromosomes are literally moving to the middle of the cell, and they're moved there by the spindle fibers. So if I were to find the middle of the cell, that would be right here. Sometimes they say the chromosomes move to the equator of the cell. If we take a look at the picture on the left, over here, you're going to notice that the nuclear membrane is gone, and I see these black things in the center. Those are our chromosomes, so those are like in our picture, our blue chromosomes all lined up. And then you'll see those red lines. Those are our spindle fibers that are attached to them. So the spindle fibers are what's kind of helping move the chromosomes around. And then you'll see that you've kind of got these anchors. These are our centrioles that the spindle fibers are anchored to. Something I just want to point out is that the chromosomes don't have to be lined up vertically in the middle, as long as they're anywhere in the middle. So like at this picture over here, the chromosomes are lined up in the middle, but this way across the cell. It doesn't matter how they're positioned, because remember, cells can have any orientation. But the idea being is that they're going to be in the middle of that cell. So the next phase of mitosis is called anaphase. And the key word here is apart. So our chromosomes were all lined up in the middle, and now those chromosomes are going to get pulled apart. So we had a double set or duplicate set of chromosomes. So if we look at our picture over here, we had identical copies, identical copy on the left, identical copy on the right. So now one set is going to get pulled to the left side, and one set is going to get pulled to the other side. So when those chromosomes are pulled apart, we're making sure that we have an identical set of DNA going to the left, an identical set of DNA going to the right. What's happening here is that the centromere, remember that was this red protein in the center that was holding our sister chromatids together. Well, an enzyme is going to break down that centromere, allowing these sister chromatids to get pulled apart. Okay, so remember, these two pieces here were called sister chromatids. Okay, so now one sister chromatid goes to the left, and one sister chromatid goes to the right. So if you can remember A for apart, that's what you got to remember. So looking at our actual picture, you can see the same thing happening here. Our spindle fibers are basically reeling in and pulling those chromosomes to the opposite ends of the poles. All right, so let's take a look at telophase. So now my chromosomes have kind of moved to the opposite ends of the cell. We're now going to start to get our two new nuclei to reform. So I'm going to show that with our dashed line. That's going to start to come back. So our nuclear envelope is going to start coming back. And so now that we're going to be about done moving our chromosomes, my spindles are going to kind of detach and kind of disintegrate. So they're just kind of not really needed anymore. And my chromosomes, they're going to start to unwind. So they're still going to be visible in this phase, but very shortly they're going to start to become not easily visible anymore. So telophase is going to kind of blur with kind of cytokinesis in a sense. So if you take a look over at the picture here, you're going to kind of see yes, we're starting to get the nuclear membrane is going to start coming back. And I've got my chromosomes. I can distinctly see I've got sets of chromosomes on one side and a set of chromosomes on the other side. You can still see the spindle fibers a little bit, but they're going to start disintegrating soon. But you're probably like, yes, I start to see the membrane pinching in a little bit. So this is where telophase and kind of cytokinesis, they do blur a little bit. So another way of thinking about this, it's a lot of the opposite things or events that are happening in terms of prophase. So like prophase, the nuclear membrane was dissolving. So now the nuclear membrane is reforming. Instead of the spindles attaching, spindles are detaching. Okay? Instead of the chromosomes condensing, the chromosomes are unwinding. So a lot of opposites here in terms of what happened in prophase. So the final process is cytokinesis. So cytokinesis is when the cell itself is actually going to divide into two new daughter cells. So in order for this to happen, the membrane has to pinch in. So you're going to see the membrane pinching in on both of these sides to get our two new cells. And remember, the whole point of all of this happening is to create identical cells. Okay, if I have a cut in my skin and I want to repair that skin that was there, I want to make identical skin cells. Okay, I'm not looking to create some new DNA. I want to replace the skin cells that were there before. So I'm making identical cells. Now in an animal cell, the membrane is going to pinch in to form the two cells. But look down at the bottom. You can probably recognize this being a plant cell because of the box-like shape, the rectangular shape. I can see the cell wall present. So the membrane can't, the membrane can pinch in, but remember, you've got the cell wall and it's too strong. So what's going to happen instead is in the middle, you're going to get something form, and that's too hard to see. I'll do it in black. You're going to get a cell plate is going to form, and that's going to turn into the new cell wall in a plant cell. So to recap, mitosis is nuclear division. And the whole point is I want to redistribute my chromosomes equally so that when my cells do divide, they get an identical set or copy of DNA in each of the two new cells. And mitosis is just a part of the whole cell cycle, where when the cell does complete the cell cycle, we are creating identical cells to replace damaged cells perhaps, or to grow.